Okay, so now that we've covered the basics of genetic inheritance, it actually puts us in a position to analyze natural populations. So natural populations, typically we mean human populations. So these are populations where we can't control the breeding. These are uncontrolled crosses. A doctor might have a patient come in and they have a disease, it might have a genetic basis, and so they aren't able to determine does this person have the disease by doing a test cross. Instead, they have to use analysis of existing individuals in order to try to figure out what's going on. And so the tool for that we use is a pedigree. So a pedigree is a description of the crosses that occurred naturally. So these are natural populations that we're studying, and these are basically just a family tree with respect to a given trait. And so there's certain things that we indicate for each individual. We indicate the sex. So a female sex is indicated by a circle, male sex is by a square, and then an unknown sex is indicated by a diamond. Affected individuals, if they have the disease or the trait, they're colored in. So here we just colored it blue. If they don't have the trait, then they are just left all white. So if two individuals have a offspring, then they are connected together by a horizontal line. So a female and a male individual had kids together. These parents are unrelated to each other, so there's a single line. And so they would then have offspring, and so the offspring are then designated by vertical lines. So if we had something like this would indicate this female and male individual had 4 children. The oldest is listed with the smallest number. The youngest is listed with the largest number. And so this couple had a male oldest child, then a female child, then another female child, and then finally one more male child. So they had 4 different children. If those two individuals were related, for example, if they were siblings, cousins, or second cousins, then we would also indicate that by using 2 different horizontal lines between them. So this would indicate not only did they have offspring, but they were also related to each other. And so for recessive diseases, for rare recessive diseases, this is often the case because it makes it more likely that these rare alleles can pair up together, is when related individuals have offspring. You can also indicate twins. So if they're fraternal twins, you have this bifurcation so that this vertical line then bifurcates into 2 individuals. So you have a daughter and a son that are twins, but they're dizygotic, so they don't have the exact same genetics. And you can also have identical twins, and so in that case you add this additional horizontal line. Sometimes if you have a large family, you don't want to indicate all of the individuals that are unaffected, and so you might sum it up together by saying there's 4 male and 4 female individuals that do not have the trait. Proband. So the proband is the first individual that meets with a doctor, let's say, that has the trait. And so this individual goes to a doctor and they have some sort of genetic disease. And then the doctor then fills out the pedigree to figure out who else in the family might have the disease. So that first individual that is seen is the proband individual. Individuals that have died is indicated by a slashed line. If you know for sure that someone is a carrier, you can indicate that with a dot. So a carrier individual would be a heterozygote, big A, little a. So they do not have the disease if it's a recessive disease. But they can pass the disease on, and then generations can be indicated by Roman numerals. So you can have both recessive traits and dominant traits, and so recessive traits are when the disease is caused in the recessive state, little a, little a. A dominant trait is caused when the heterozygote also has the disease, or of course if the homozygote has it as well, they would also have the disease. But typically for dominant diseases, the genotype is almost always big A, little a. So these are just some common examples of traits and diseases: Tay-Sachs, sickle cell, phenylketonuria, color blindness, cystic fibrosis, and then these are examples of dominant traits. Huntington's disease probably being the most famous of the dominant traits. So here's an example of 2 progenies, pedigrees, one for an autosomal recessive trait and one for an autosomal dominant trait. And so our goal typically is to predict the genotype of individuals based upon analyzing a pedigree. And so how can we do that? So if we know that this is an autosomal recessive trait, we can fill in the genotype of all the affected individuals. So this individual must be little a because they have the trait. This individual right here must be little a because they have the trait, and so forth. So this is the proban, this individual. This individual has one sibling that has it and 4 other siblings that do not have the disease. This individual has 2 parents, a father and a mother. They do not have it. The father's father has the disease. So this would be, for example, the aunt and the uncle of this proban individual. This individual also has 2 aunts and 2 uncles on the mother, 1 aunt and 2 uncles on the mother's side. So, all right, so we've managed to fill in 3 individuals. Is there anything more we can do? And in fact, we can. So we can also follow this information in 2 directions. We can either follow it through the offspring or follow it through the ancestors. So how do we do that? So an example of how we do that is this individual is little a, and so we know for certainty that this male individual must produce a gamete that's little a. And so that means that all of these individuals right here must be carriers of the disease, okay? Because they must have received the little a from them. They do not have the disease, so they cannot be little a, because if they're little a, then we know they would have the disease. Then we can also follow it in a different direction right here. So this individual right here, they for sure received a little a from their father, but they must have also received a little a from the mother. So the only possible way they could have the disease is if they received it from both of their parents. And so again, we know that this individual is for certain a carrier because this individual was able to have offspring with the disease, but they did not actually have the disease. We also know that one of these 2 parents, so if we follow it in this direction right here, we also know that one of these 2 parents must be a carrier, but we can't tell from this information if it's either. So either this individual is a carrier, this individual is a carrier, or potentially both individuals are carriers. We know we can actually calculate the probability of that happening though. So, okay, so these 2 individuals are carriers. So now we can also ask the question, what is the genotype of this individual? So if we had a if we had a So essentially, if we had these 2 individuals right here and we asked, what is the probability that their offspring would have the disease? So we simply follow the Mendel rules that we saw before, and we know we get a 1-3-1 ratio. So 1/4 of the population would not be carriers, 1/2 of the offspring would be carriers, and 1/4 would have the disease. So from these 2 individuals, we expect that 1/4 of their progeny would have the disease. So here, 2/6 of them, 1/3 of the individuals have the disease. So these other individuals, we can ask, what is the probability that they're carriers? So you might look at this individual, and this is where it gets a little bit tricky to do these pedigrees. You might look at this individual and think, okay, the probability that this individual is a carrier is 1/2. because if we know we have 2 individuals that have it, then the probability is 1/2. But you can't actually do that because we now know extra information about this individual. We know for certain that this individual is not little a. And so we basically we know that the probabilities have to add up to 1. And so the probability that this individual is a carrier is not 1/2, but it's actually 2/3. And so how do we do that? So we know that this individual is not a recessive, does not have the trait, so they're not little a. So if we add up the remaining probabilities, it's equal to 3/4. And so we have to renormalize what's called renormalization. We renormalize these probabilities to take into account this extra information. So 1/2 divided by 3/4 is 2/3. So this individual has a 2/3 probability of being a carrier and a 1/3 probability of not being a carrier. And so now we can add these up together. So what about a dominant pedigree? So again, we know for a fact that all the individuals that don't have it are little a. And so that would be, this individual, this individual, this individual, this individual, this. And then that would mean that this individual would have to pass on a little a to all of them. So all of these other individuals are big A, little a, big A, little a. So dominant traits are usually a lot less interesting to analyze, and they're all pretty straightforward. So these are some of the questions that we would want to answer. Just think of us as like human doctors. And so we want to be able to answer, like, what individuals do we know for sure are carriers? What is the probability that this individual could pass on this disease to another individual, etc.? So hopefully all that makes sense. You'll have some activities that really hopefully help you understand.